leading mass atomic imaging centers in the world. Huge center that I didn't ever hear, so hear about much. But he obtained his Bachelor of Science and Master's in the University of Antwerp in Belgium, a PhD from the University of Nottingham in the UK, and then did a postdoc in the French Center for Scientific Research. He was also a postdoc at Penn State University, and I met him by luck, fortunately for us, in a meeting with the Chang Zuckerberg project. And so he made his introduction and said he worked in ToF-SIMS, and Wagner and Gilbert and I were, at the time, very interested in getting a system, so we started talking and collaborating. And he worked exactly in this area with biological material. We met again in San Francisco and spent several hours, and then he started making more and more connections. So it was advantageous for both of us, and he's helping us a lot at the moment. He introduced us to a friend of his who's Brazilian, who's at the National Physical Lab in the UK, very well-known young fellow. And he also works on ToF-SIMS, and Gustavo Trindade. So now we're working together with these two, and they're guiding us in which instrument we need, which sort of accessories, and helping us a lot. So this is something very, very important, because these instruments on the order of 10 to 15 million reais each, so it's important to select the correct one. So we'll discuss next week. We go to a meeting, and so with Sebastiaan, Gustavo, and representatives from companies, we'll discuss more details about what's the best for UFMG. Interestingly, Maastricht University has a collaboration with UFMG and the Project Usuara. I have a student, a joint student, that just finished her PhD here recently. Thomas Clays was here a couple of weeks ago. He gave a seminar and visited people. So there already exists a good collaboration with Maastricht. So with that, I'll let Sebastiaan tell you about his work.
All right, thank you very much for the introduction, Greg. And yeah, let me start off by thanking Greg and everyone from the Microscopy Center for the invitation that I'm able to give a seminar here for you all today, as well as for picking me up at the airport yesterday. That was greatly appreciated. And it is quite funny how a seemingly unimportant table session at a CZI meeting in San Francisco led to where we are today. So it's a nice example of a butterfly effect. So the talk of my presentation is an anthology of integrative ToF-SIMS imaging. ToF-SIMS stands for time-of-flight secondary IMS spectrometry, and we'll go into a little bit more depth as to what that means. But the first part, anthology, simply means that I'll be presenting a collection of stories of different research lines that are currently going on in my group, because the UFMG is interested in acquiring one of these instruments, and I just want to be able to show you what kind of research is possible with such an instrument. And hopefully we'll be able to show that quite diverse research is possible. Just within my own group, there are already very different topics that we're looking at. Greg already introduced my background, but now there's also a visual aid. So I grew up in the city of Antwerp, in Belgium over there. Got a bachelor's and then a master's in chemistry, and that's also where I first got acquainted with imaging mass spectrometry in the laboratory of the now, unfortunately, late Professor Luc van Vaak. And after that, I figured, let's see a bit of the world, and I continued on doing a PhD at the University of Nottingham under Dr. Misha Zeltzer. And that's where I made the switch from using ToF-SIMS for materials science to using ToF-SIMS for basic biology. So I did a PhD thesis on the three-dimensional imaging of neuronal networks of primary neuronal cells of rats. And then after that, I moved over to France, to the French National Center for Scientific Research in Paris, where I worked with Dr. Alain Brunel. And that's where I started working more and more with clinical applications, where I got to know physicians interested in learning more about disease mechanisms using these types of techniques. And then I moved over to the other side of the ocean, and first started as a postdoc at Penn State, and then after a year negotiated myself to a assistant research professor position. And that's where I really became more of an independent researcher, where the grant writing finally started kicking in. And then after that, I got an offer I couldn't refuse, and started a tenure-track assistant professor position at Maastricht University. And on the left, you can see me in December 2020, in the middle of the pandemic, starting at the Faculty of Science and Engineering. If you want to have some more information about the stuff we do, that's the website over there. And I started out with a double appointment. So on the one hand, I'm part of the Faculty of Science and Engineering. That's where also most of my teaching occurs, so that's where most of the teaching load is. But the research group is based at the MFRI Institute in a different building, the US 50 building at the Antwerp campus. That's also where my office is and where my research group is based. So teaching in one location, research in the other. MFRI, some people think it stands for M41, but it's actually MFRI. It stands for Maastricht Multimodal Molecular Imaging Institute. And it is in fact the largest molecular imaging center in Europe. We are composed of two separate divisions. Here we go. So on the one hand, we have the, here we go. Yes. So on the one hand, we have the division of endoscopy. So those are the people that occupy themselves with various types of electron microscopy. And I am part of the division of imaging mass spectrometry. And we have multiple PI groups in each of these divisions. Here's mine over there, which focuses on target pathology, mainly using time-of-flight secondary ion mass spectrometry. So we could also call it the ToF-SIMS group, if you like. And the institute is run by Professor Dr. Ron here. And then the endoscopy division is run by Peter Peters at the moment. And there's a dual position where Ron also is the director of the division of imaging mass spec. But that's sort of the environment we're in. So I'm only one PI in a much larger pool of PIs at this Multimodal Molecular Imaging Institute in the Netherlands. And all the different PIs have their own focus. So for example, my colleague Ian Anthony, that I will present later on, really focuses on instrument development. Mainly SIMS instrument development, so the two of us work very well together, where I'm the applications guy and he's the instrumentation guy, and he needs my input to design his instruments, and then he needs me to actually go and test his instruments. So it's a nice collaboration that we've got going on. Benjamin Belk is mainly focused on imaging bioinformatics. We have our core facility manager, Michiel van der Bos. We also have Rob Freken, who's mainly working with pharmaceutical companies. Eva Kuipers, who's mainly interested in diagnostics, but intraoperative diagnostics. So it's all about live imaging mass spectrometry on the spot during surgeries. Martin Boney is more interested in systems imaging, and then mine is more target pathology using SIMS, so trying to understand disease mechanisms, finding biomarkers, that kind of research. So, just by a quick show of hands, who knows what ToF-SIMS is? Who has already heard of it? Okay, a few hands. Who has already heard of imaging mass spectrometry? Okay, a few more hands, that's good. Who has heard of mass spectrometry in general? Okay, still not all hands, so we'll start from the beginning, real quick. And I was thinking about what to do about this, because I don't want this to be a talk about the basics of ToF-SIMS, because I'm sure you'll have plenty of those lined up in the future. So, for the moment, just consider that mass spectrometry in general is just a way to get information about molecules. And this is an example of a mass spectrum, and you basically measure the abundance, the intensity of your molecular ion over here after you give that molecule a charge. Giving that molecule a charge also gives it energy. That energy can then cause it to break apart according to logical pathways. So it will break apart in a specific manner because of the structure of the molecule. And that then gives you a very characteristic fragmentation, which means that you can fingerprint these organic molecules. So it's a highly specific way to identify them. Quick question for the undergrads. Does anyone know what this molecule is? Very good, it's cocaine. It's good that no one knew. How do we create a mass spectrum like that? We need a mass spectrometer, and a mass spectrometer is very simple. It just consists of 3 different components. You have an ion source in which you ionize, give your molecule a charge, and where it can fragment. Then you need to separate those ions according to their mass in a mass analyzer, and then you detect them. And if you have those 3 components, an ionizer, an analyzer, and a detector, you have a mass spectrometer. And in the case of ToF-SIMS, the SIMS part stands for secondary ion mass spectrometry. That's the ion source part. The ToF part stands for time-of-flight mass analyzer. That's the mass analyzer part. And in our particular system, we use a multi-channel plate detector. So that's all what ToF-SIMS really is. SIMS is the ionization mechanism, ToF is the analysis mechanism. And that's it. So that's a mass spectrometer. What is an imaging mass spectrometer? We usually abbreviate this as IMS or MSI, although I recommend using MSI to avoid confusion with ion mobility spectrometry. This basically visualizes the spatial distribution of your chemical compounds via these M over Z ion intensities. So you can think of this as a 2D image or a 3D image where every pixel or voxel is a full mass spectrum. So you end up with a hyperspectral image. And then you can isolate a particular ion you find interesting and show how it's mapped on top of your surface. There are 2 big techniques that allow you to do this. The first one is secondary ion mass spectrometry, the one we'll be discussing today. And the other one is matrix-assisted laser desorption ionization. And apart from that, you also have a bunch of other ones, ambient source like DESI, LASI, MELDESI. But the 2 big ones are SIMS and MELDI. And this is the one that we have over at Maastricht. So we have one from a company called Physical Electronics. And this is an ToF-2. And the way it works over here, we have our primary ions, and that's also where the name of the technique comes from. It's secondary ion mass spectrometry. Why? Because we use primary ions to generate secondary ions. So we shoot primary ions at our solid surface under a vacuum. That impact of the primary ion causes a collision cascade and causes the top layer of the sample to come off, desorb, and ionize. So the impact generates secondary ions from the surface. So then we have a packet of ions right above the surface, and to use an electrostatic potential to accelerate them into the time-of-flight mass analyzer. And all we really measure is the time of flight. So we give all the ions the same kinetic energy by accelerating them with an electrostatic potential. And because they have the same kinetic energy, but different masses, 1 over 2 mv squared, they will have different velocities. So the lightest one will arrive at the detector first, the heaviest one will arrive at the detector last, and then we just convert time into mass. And that's how we generate a mass spectrum. And then finally, how do we get to the imaging part? Well, we are able to focus these primary ion beams down to a very narrow diameter. 1 micrometer diameter, half a micrometer diameter, sometimes even down to 50 nanometers. And because we have a very narrow primary focused ion beam, we can measure mass spectra, secondary ion mass spectra, spot by spot. So basically rastering our entire sample surface in 2D, and that's how we can reconstruct a 2D image where every pixel is a mass spectrum. We can also do this in 3D, because we have other types of primary ion guns, so we can also use argon GCID or C60 gun. And in that case, we can remove layers of material, take another 2D image, remove a layer of material, take another 2D image, and then you have a 3D reconstruction, where every voxel is a mass spectrum. So that's it in a nutshell. So if you aren't entirely sure what I've been talking about, just imagine it's a microscope, but instead of looking at light and optical density and colors, we're looking at molecules. So in essence, that's really what it is. It's a microscope that can look at molecules. has a similar spatial resolution, but it's able to look at molecules without labeling them. So you get your molecular information without any labels just by measuring their masses. Our particular instrument is equipped with multiple of these ion beams. So we have a liquid metal ion, gamma bismuth, LNIC for mainly used for imaging. We have a C60 gun we can use for sputtering and imaging as well. And we have an argon GCIB cluster gun that we can use for sputtering. We also have charge neutralization modules, so we have a flood gun. We also have a argon gas gun that we can use for dual beam charge neutralization. And we also have a MS2 system, so tandem MS, very simply put, we can isolate one of the ions that we send to our detector. So let's say that we find a mass that we find interesting in our mass spectrum, but we don't know what it is. We can isolate it over here, bunch it into another cell where we refragment it, and then send it to another linear time-of-flight detector. And in that case, we get a mass spectrum from an ion in a mass spectrum. And that allows us to then uniquely and unambiguously identify masses that we have in our spectrum. And because of the way this instrument is designed, we can also do this in an imaging format. So we can look at tandem mass spectra ions in image format as well. And this was actually the very first such instrument, because together with physical electronics, this was actually developed at Maastricht. So this was actually the very first of these instruments. So let's move over to some nice examples. So our first nice example is this little paper that we published last year. And this is the work of Kimberly Garcia, my PhD student. And it was a nice side project together with Dr. Birgit Blom, who is an inorganic synthesist who is interested in making drugs. And he made this particular one, a arene-ruthenium complex, that you can see on the left-hand side over there. And this seems to have some anti-cancer properties. So it's anti-neoplastic and anti-metastatic. But you need to confirm its structure, which is quite difficult doing electrospray ionization, because you don't really have a lot of protonation sites on this molecule. So he asked me, over a beer, can you put this in your instrument and check if you can confirm the structure of this molecule, because no other mass spectrometer has been able to do it so far. So then I turned to Kimberly and told her, here's something new for you to do. So, there we go. And this turned out to be a very nice example of the tandem MS feature in this particular instrument. So we were able to look at each and every single ion in the first mass spectrum, the MS1 spectrum, and then obtain a second mass spectrum of those individual ions. And then we were able to figure out which peak fragments into which peaks. So this allowed Kimberly to figure out the entire fragmentation pathway all the way from the largest ion down to the smaller ones. And after a little bit of puzzle work, she came up with the following fragmentation mechanism. So the tin chloride splits off as a negative. You see that in a negative polarity, and then you have loss of several groups through alpha scission over there. And the one thing that was quite interesting was this substitution by oxygen from water in the solvent. Don't ask me why, but it had something to do with the activation properties of the particular drug. So Birgit was very happy to see this, even though we didn't really understand why. But yeah, so the tandem MS feature allows us to very neatly separate out all the different fragments, figure out where they are from. And the mass resolution of that instrument, when we zoom in on the particular peaks we look at, we can also see all the isotopic patterns of the different elements that are present in that drug. So whenever there's a ruthenium present, give a very specific isotopic pattern. And the blue arrows show you the theoretical distribution, and in almost all these cases it was a near perfect match. So apart from looking at the actual masses and the mass accuracy of the peaks that we identified, the fragmentation pathways that we identified using the tandem MS feature, we were also able to look at the isotopic distributions for additional confidence. So in this case, we were near, this is 100% confirmation that the drug that they synthesized was actually the drug that they synthesized. And this then led to another nice development, because as it turned out, we get very strong signals for this drug, both at the elemental but also at the molecular level. We're now going to start a project where we're going to expose zebrafish to these drugs to see how these drugs actually metabolize in living organisms, and then use the time-of-flight secondary mass spectrometer to see, with the imaging modality, where the drug is located in various tissues types. So this is already one example of how SIMS can be used. It is really good for looking at organic molecules. So if you're interested in extracellular materials like drugs or toxins, SIMS can be a very sensitive way of observing them. Same thing with metal ions that are present. Then on to a second story in our anthology, and this is a pilot study also spearheaded by Kimberly. And this is in collaboration with two people from the Ludwig Boltzmann Gesellschaft, Dr. Nikolai Brodnik and this postdoc, Nadja. And they are doing an interesting study where they're trying to look at senescence, or cell division, or cell longevity. And one way of doing that, or one way of figuring out how fastly cells divide, is by feeding them with some kind of isotope, in this case nitrogen-15 enriched food. And then when they are born, so you give it to a mother mice as she is pregnant, so that the fetuses are completely saturated with nitrogen-15. And then after they are born and you take them off the feed, cells that divide with normal feed that contains normal nitrogen will lower in their nitrogen-15 content by half. And then when it divides again, will divide again by half. And then you can see the difference between cells that divide quickly and cells that don't divide at all. So for example, neurons don't really divide all that much, so we don't expect a big decrease in the brain. However, in skin, you have a lot of rejuvenation of your cells over there, so we expect to see a big decrease once we take them off the feed. And the same thing with wounds. So if you have a piece of tissue over there and you wound it, after you take the mice off the feed, you expect all of those cells to be new cells or newly divided cells. However, how do you observe those? Because they had read about these experiments, but how do you actually visualize this? That's how we ended up with our time-of-flight secondary mass spectrometer again. So they gave us a couple of first samples to look at, and these are the analysis parameters that Kimberly used. And these are the results of the pilot study. So these are the mouse tissues right after the pups were born and then sacrificed. So in that case, we had a look at both liver and skin tissue. And over here you can see some images of what the skin tissue looks like. So you have several layers where you have the epidermis, dermis, hypodermis. And we were able to compare skin tissue from a normal mouse together with one that was fed the nitrogen-15 enriched food. And when you have a look in the mass spectra, you quite clearly can see that in the enriched tissue you have a very big peak over here at C15N-, which is a very low, a very fragmented ion in the spectrum, but the one that was most easy to identify. And moreover, it has a very low background. So we could have a very quick look at the difference between the CN- fragment and the CN15N- fragment, and see what the theoretical enrichment was. And we could do the same thing with the CNO fragment over here, and both of these are typically peptide backbone ion signals. And when you have a look at the ion image, so over here you can see normal tissue, so you can see some oxygen, the CN- fragment, the total ion counts, and then when we look at the C15N- fragment, it's blank, because it's not enriched, it's not supposed to be there. And in the enriched mouse skin tissue, you can see it's the same image that you would get for the CN- image over there. So this is exactly what we need to then see the division of cells going forward in the future. So then we basically be comparing how cells evolve from complete enrichment all the way to complete depletion, depending on their age. We could do the same thing in the positive polarity. In that case, we mainly had a look at lipid fragments that we can attribute to the phosphocholine head group. But over here, you can probably notice that there was a bit more of a background peak. Same over here. So in that case, you do see a little bit of a background in the tissue that wasn't enriched. So in that case, our conclusion was that we probably should stick with the negative polarity and use the ions that have the lowest background to create the best images. There you go. And in conclusion, I think we ended up determining that for the negative mode, we were able to see about 44% of uptake of this nitrogen-15 by the fetuses. And now that we have done the proof of concept, we now know that we can do this. Now we're going to move forward and do the actual study, where we're going to have a look at wound healing as well as several different tissue types to see how quickly cells regenerate. And this we were also able to combine with traditional immunofluorescence staining. So this is actually quite neat. We took the tissues out of the SIMS instruments, so they are completely dry. It's an ultra-high vacuum environment. So they are completely dehydrated, and we sent them back over to Ludwig Boltzmann. So that's quite a harsh treatment of tissue. And over there, we were able to rehydrate them and apply a immunohistochemical staining and obtain the following images that are a very nice match with the ToF-SIMS images over here. So now we're able to combine traditional immunofluorescence imaging with our ToF-SIMS imaging as well. And when you have a look at the TAPI stain over here, the nuclei in blue, they very neatly match these phosphate ions that we see in this image. So this is another example of something we can do with SIMS, and now we'll move over to a separate investigation that has been going on, also involving mouse skin, so there's at least a little bit of an overlap there. And this is actually a project that has started back when I was a postdoc at Penn State. Over there I got talking to Professor Neil Christensen, Professor Yu Sheng Zhu, and Dr. Chia Van Hu. And they were investigating human papillomavirus, which is a pretty severe virus because it causes about 5% of all human cancers. The most well-known one is probably cervical cancer, and roughly 270,000 women die every year of cervical cancer. Although weirdly, lately, oropharyngeal cancer cases amongst young Caucasian men are increasing and actually overtaking the cervical cancer cases. And there's really no information as to why that particular subgroup is being hit by this particular pathology. And that actually leads us to the reason for the research. So in general, when it comes to tumors caused by human papillomavirus, there are really no precursor lesions. It's really difficult to see the developmental stages and the disease progression to malignancy with this particular pathology. And there's also a lack of mechanistic insight, so there's really no idea of getting to a theranostic agent at this moment. So we started thinking, and we were wondering, can we distinguish normal and tumor tissues using time-of-flight, secondary mass spectrometry, and can we have a look at the following targets? One are the lipid profiles, and they're considered quite important molecules in tumorigenesis. For example, the importance of ceramides has been established in cancer. Another one is energy metabolism, so we want to have a look at the glucose citrate ratios, because it is known that this is very different in cancer cells compared to normal cells, and has not yet been investigated in papillomavirus-induced tumors. And comparing blood serum of animal models also showed that these particular metals are quite different in concentration between animal models with cancer and animal models without cancer. So these are the targets we're interested in, and these are all targets that we can look at with ToF-SIMS, because ToF-SIMS is quite good at imaging certain things. It's really good for looking at elements. You can look at inorganic molecules. You can look at organic molecules up to a mass of about 2,000. So you can look at lipids, you can look at metabolites, you can also look at smaller fragments, you can look at extracellular materials like toxins and drugs, But anything above M/Z2000 is a little bit difficult. So you cannot use it to look at proteins or DNA or anything like that. But for these targets, ToF-SIMS was actually a very good system to use. And hopefully it could replace the current golden standard, which is histology, but that is not very sensitive, also not very specific, and you cannot do any precision imaging. So we got started, and we got ourselves 5 healthy mice and 5 mice exposed to a mouse papillomavirus. Section these tissue blocks in the cryostat, making sure to use OCT to hold the block in place, but not allow it to cover the tissue itself, because OCT masks ion signals in SIMS, so you do want to keep it away from the area that you want to image. Sort of normal sectioning, 12 micrometer sections, minus 20 degrees. We then tall-mounted all of these on conductive microscope slides. So this is one thing that is important. Whenever you put a substrate in the SIMS, it needs to be conductive, because we use an electrostatic potential to send it into the mass analyzer. So we coat all of our glass microscope slides with indium tin oxide, which is transparent, so you can still do all of your normal microscopy imaging, all that kind of stuff, but it is conductive. Then we acquired some H&E images as well, so we always get serial sections, and we use a couple of the sections in the series for SIMS imaging, and a couple of the sections for H&E imaging, and a couple of the sections for normal immunostaining. We acquired both positive and negative polarity ion images, because in SIMS you create both. The primary ion impact creates secondary ions that can be both positive and negatively charged. But we can only use one type of electrostatic potential to extract them, so we need to acquire both of these in separate experiments. And that's a nice example of what the data looks like. So because you get your image over here, there's a total ion mass spectrum, so the total ion image over here, and every single pixel is a full mass spectrum like this. And this is the positive polarity, this is the negative polarity. So in a sense, it's actually the same data set, we just get different types of ions out of it. So if you add everything up, we have 2 classes, healthy and unhealthy. We have 5 mice, 2 polarities. We also had 5 repeats per tissue section, so we have about 100 of these data sets. If you combine the positive and negative polarity, then we still end up with about 50 unique image data sets. And that's a lot of data to analyze. And this was also just as I transferred over to Maastricht University, but I told my collaborators, don't you worry, the moment I get settled, I'll get back on it, start analyzing those data sets. So 2 years later, I hired a postdoc, and then we finally could start working on analyzing those data sets. And the postdoc in question is Maria Shamareva, on the left over there, and 2 master students also joined the project because they took my course on advanced image processing and artificial intelligence. And even though I told them that if they wanted to become interns, it would be unpaid, it wouldn't increase their grades, and it wouldn't give them any course credits, they still wanted to do it for some reason. And this is a first glance at the data, and the lighting might not be ideal because we use some color coding over here, but we basically labeled several certain ions in a red color, certain ions in a green color, certain ones in a blue color, and then you see the overlay on the right. So you can see your cholesterol sulfate, 24 carbon fatty acid chain, a PE fragment, and then when you overlap these images, you can see that they actually start separating into different layers. And those layers correspond to what you would expect for the different layers in the skin, where you have multiple layers. Yeah, that looks a little bit better. Can everyone see this? Yeah? Okay. So yeah, so we can see that different molecules organize themselves along different spatial distributions. And those spatial distributions match the different layers in our skin. So the cholesterol sulfate usually matches a layer in the epidermis. The fatty acid over there matches a layer just above it in the stratum corneum, and the PE fragment is present in the dermis all the way down into the hypodermis. So just by looking at a couple of these molecular fragments, we can already start to see how layers separate themselves out. However, what we want to do is compare healthy versus unhealthy tissue. And as the ion images over here show you, there are different layers over here. So we cannot really have a look at the total mass spectrum of one image data set and then compare it to another one. We actually need to do some image segmentation in this case, because you want to make sure that we compare healthy epidermis with sick epidermis, that we compare healthy dermis with sick dermis, and healthy hypodermis with unhealthy hypodermis. And there's also a background, so there are multiple different pixels over here that can be organized into different cell types or backgrounds, etc. So the first step is an image segmentation, so we can finally do a pairwise comparison. And this might be going a little bit into detail over here, but this has to do with the data structure of ToF-SIMS images. So apart from knowing how to operate the instruments and knowing how to prep your samples and interpret your data, you also need to understand how to analyze your data. So you need to have people with quite varied skill sets in order to manage these types of projects. So one aspect of SIMS data is the fact that its noise structure is not normal. It is actually governed by a Poisson distribution because we're using counting statistics. We're counting the ions as they arrive on the detector. In that case, you get a Poisson noise over there. Now, that isn't a problem if your signal is very high, because then the Poisson distribution starts to approach a normal distribution. But if the counts are low, it is very different. And that's actually something you get with SIMS images, because topography can also influence the secondary ion yield. So over on the left you have the total ion images, and on the right you have the same total ion images but thresholded at 10 counts per pixel. And you can see that all those white areas are the ones that are removed from the data set simply because there are not enough ion counts in those pixels. And then we got working on a data analysis pipeline. So we start off with the raw data sets. First thing that you have to do is calibrate it using known ions in your mass spectrum. So we always use an internal calibration. Then, because we have different mass spectral bins in our data sets dependent on the time to digital converter, so we actually measure at certain time intervals every 128 nanoseconds, if I'm correct, are binned as a mass spectral interval. But then you end up with a very high number of intervals that is a little bit too much for processing, so you want to downbin them a little bit. Then, because topography and primary ion intensity of the beam can also vary over time, you want to normalize all of your images to the total ion counts per pixel to take away any instrumental variation in your measurement and between measurements. So basically to make them more comparable. Then you probably don't want to use those parts of the signal that just contain noise. You want to have the parts of the signal that contain actual signals, so you need to come up with a peak picking algorithm. And then you can play around with the signal-to-noise ratio, the amount of background, the minimum number of counts per ion peak. So that also introduced a little bit of user bias at that point, because these are decisions that the user has to make. And then finally, the scaling, where you have to take into account the noise structure of the data. Although in this case we do have an advantage, because when you use the normal standard deviation, which is biased, because you do it over all the different pixels, you get a very high number of observation data sets, and then the standard deviation actually approaches that of the Poisson distribution. That's probably a little bit too much detail. But yeah, then the next step that Maria got on with is some dimensionality reduction, because we're looking at a couple thousand mass peaks for each of these data sets. So we wanted to reduce that amount of data by doing a principal component analysis, creating linear combinations of these variables. And then to get the segmentation going, we performed a k-means clustering on the principal components of the mass spectrometry images. Let's see. Well, these are some additional details about those different steps. And for time's sake, I think I'll skip over these. This shows the importance of scaling, because you want to have a distribution centered around 0 with the same standard deviations as before and after. And these are some of the results of the PCA, where you can already see that the PCA is separating out different layers within the skin tissue. But the really neat thing is, if you're looking at the PCA, you actually need to put in some time and effort to understand the signals, and there can be some convolutions. When you do it by k-means clustering, you actually get a very nicely clustered set like this. And see if you can, so over here you can see how it very neatly separates out the background, low intensity region, epidermal region, region in between, and then the dermis itself. So without too much user input, although it was quite a bit of work to get all of this done, just by looking at the chemical data using PCA and then clustering the pixels using a k-means, you can separate out all the different layers in your skin tissue. So this is exactly where we want it to be. We want to be able to separate out all the different layers so we can do pairwise comparisons. And I think the point of this slide was the results we got when we first started 6 months ago or 9 months ago and where we are now. So it took a little bit of time to actually optimize all these parameters. And then for the pairwise comparison, we're going to use some supervised machine learning. So in that case, we're going to use a random forest algorithm for the classification. And the nice thing about random forest, even though this slide is a little bit complicated, is that it allows for quite an easy classification because it thresholds each variable to get very pure subsets in the tree. So it is quite efficient when you calculate the classification results for new data. And it also gives you results for the predictor importance. And I think I'll skip over a few of these. Okay, here we go. So after the classification, so we had a classification with a very high classification rate. So we were able to separate out our diseased from a non-diseased tissue, not by labeling, but by processing the data using the random forest that we built up with the algorithm. And then we were able to figure out which particular ions were the most important ones for that. And you can see that several ions in the high mass range, which corresponds to intact lipids, are important in the decision-making process. So those probably have something to do with the disease mechanism. And now is the time where we go back to the people from Penn State, and we tell them, okay, this is our segmentation. These are the ions that we find that are different between healthy and diseased. Now can we use your expertise to get some insights in the disease mechanism? Are there any other targets that we should now investigate based on these results? So this was also a very nice example of how it can be used for biomedical research, and specifically looking for biomarkers for disease mechanism. And it also highlighted a different aspect of ToF-SIMS imaging science, which can also involve quite a bit of data analysis. Okay, next story in the anthology. So the thing that I mentioned earlier, SIMS is really good at detecting certain things. It's really good at detecting elements. You can look at inorganics, you can look at organics up to 2000 Dalton. But we cannot look at proteins. And in the very first example, we had to take the tissues out again, rehydrate them, do an immunohistochemical staining. But the downside of that is it's more work, and you can only stain a certain number of proteins at the same time, because otherwise your fluorescence bands start to overlap. So you can stain like 5 or 8 different targets at a time. Now, because SIMS is really good at detecting elements, what if we label those antibodies using elements? In that case, we can label them with as many elements and isotopes as there are in the periodic table, and we should be able to detect all of them at the same time. And there has been some proof of concept by Angelo et al. In 2014, as well as a couple other systems that have been developed, and they were able to look at 40 to 100 multiplexed immunohistological stains at the same time. And that is something that we also want to do. Now the difference is the people doing this used a nano-SIMS type system, so that uses a DC beam, we use a pulse beam. So what the main project of Kimberly is actually trying to make this work on a time-of-flight secondary ion mass spectrometry system. So we can then combine the protein maps with the lipid maps and the metabolite maps and the chemical element maps at the same time, so that we get a comprehensive overview of the different omics in our tissue types. And the way that she's going about it, she's currently sectioning fresh frozen tissue, human tissue. We're always doing an H&E stain. Start with a normal SIMS imaging experiment, just to get all the normal SIMS information that we would get from such an experiment. And then we take them out, rehydrate them, and apply our metal-labeled antibodies over here. And we have to do it in that order, because once you do the immunohistochemical staining, you quite often use alcohol washing and alcohol drying, all the kind of stuff, and that removes all the lipids. So you have to do the lipid imaging first, and then the protein staining. And that is still ongoing work, although the first results are finally starting to get in. And related to this project is actually the reason that Greg and I met, the CZI project that we started. This is in collaboration with my colleague, Dr. Ian Anthony, who is the Instrumentation Development Group. So he's the PR of the Instrumentation Development Group. And this is his brainchild, the fast mass microscope. So basically, this is a SIMS instrument that is able to measure things a whole lot faster. So all the commercial SIMS instruments use a microprobe tactic, where you shoot your focused primary ion beam at one spot, record the mass spectrum, move on to the next one. What he's doing, he's using a defocus beam, generating all the ions at the same time, so that the image in its entirety comes off, moves through the mass analyzer, and then hits a camera detector. And in that case, you can work a lot faster. So they've already demonstrated acquisition speeds up to 100,000 pixels per second. So just to give you an idea, on the Phi Nano ToF-2, you measure at about 400 pixels per second. So they were able to image the entire microscope slides in about half an hour. In a commercial SIMS instrument, that would take about a year. This is a prototype. There are still a lot of things that need to be optimized. And that's why we hired two postdocs on the CZI project. On the one hand, Maria, the one that has been leading the data analysis project. So I want her to optimize the SIMS IC protocols that we've been optimizing for the NanoToF instruments and adapt them to this instrument. And the other postdoc, Ian Sandstrom, is working on optimizing this instrument itself. And over here you can see the, let's see, yeah, so this is the TRIFT system. So it's actually the same type of mass analyzer that we have on the NanoToF system. But then on top of that, you have the time-peak street camera over here. And this is the thing that allows you to get ion images directly onto your detector instead of doing it pixel per pixel. And here you see it. So this past year she's been working on figuring out the topology tolerance of the methods, because the bigger the field of view, the more topological differences you might have because of tilt. So we want to try to minimize those, or at least figure out what amount of topology we can tolerate. She's also been working on incorporating encoders for more precise positioning and increasing the analytical speed. Well, yeah, these are some very early tests where she's been looking at gadolinium on tissue. So that's been sprayed with gadolinium on the top half, not on the bottom half. And you can quite neatly see the distribution over here. And this 1 by 1 millimeter image only took 37 seconds to acquire, which again would take half an hour with a normal SIMS instrument. So, topography tolerance, as it turns out, it is quite good because of the way that this instrument is designed. So with a normal SIMS instrument, a topography difference of about 25 micrometers already creates some shadowing. And if you go higher than 100 micrometers, you completely lose your focal plane. And with this particular instrumental setup, you can go up to about 500 micrometers. So that's a very good limit, especially if you're looking at larger areas. She's also been working on the encoders. So on the left you can see how the coordinate system worked before. So that's supposed to be a TEM grid. And on the right you can see the TEM grid with working encoders. So that's made quite a big difference. And then most importantly, she's also been working on increasing the speed. And it's probably difficult to see, but this is the speed at which it actually records these images. And she's been able to increase the speed up to about 5 million pixels per second. And what we can see over here is a slide with a piece of lemon, a nickel TM grid, some holmium, copper TM grid, a fingerprint. And then by isolating these ions in the mass spectrum, you can neatly see where all of these different parts are localized. Now, the reason why you want to combine Ian's technology with the combination of SIMS and immunohistochemistry is to become truly competitive with immunofluorescence, because we have the advantage that we can multiplex, we can look at a lot more stains, but a traditional SIMS instrument is a bit slow and only looks at a very small spot at a time, 500 by 500 micrometers. But with this, we get rid of the throughput issue, and we also get rid of the limitation in field of view. And in that case, we can look at a lot more labeled antibodies in a much faster, at a much faster speed over a much larger field of view. So at that point, we really become competitive with immunofluorescence, and hopefully we can relegate immunofluorescence to the annals of history. That was a little bit arrogant, but that was a funny joke. And I know I'm coming up a little bit short on time, so let's go to the final story in the anthology, and that is the project of postdoc Caroline Bouvier. So she and I applied for a Marie Curie postdoctoral fellowship so that she could come and join the group. And this is on a completely different type of object. So up until now it has been mainly biomedical applications. Caroline wants to work on cultural heritage, but using exactly the same methods, because as it turns out, a painting is composed of stacked layers, and it will contain a paint, a binding material, and some sort of solvent, which then evaporates. But you basically end up with layers that are a couple micrometers in size. You will have inorganic pigment particles in there of a couple micrometers in diameter. And the binders are typically oils, and there could also be glues and other proteinaceous material used to basically hold it all together. So when you start to think about it, you need to have some kind of instrument that allows you to look at the microstructure of a painting, that can look at elements, inorganics, and lipids. So we end up with SIMS again. And the only thing that's missing are proteins, but that's something that we're trying to solve in the group at the moment. So that's why Caroline was very interested in joining the group, to basically extend the protocols that Kimberly and Maria are developing to cultural heritage. Okay, there we go. So why is this important? So the artist has its intentions, they know what they want to paint, and it usually has a meaning. And they have a very deep knowledge of the physical and optical properties of each layer, how the different ingredients work together. So that creates your structure and composition, and that creates the aspect of the painting, which is then, of course, deteriorated by degradation and aging over time. And by figuring out the microstructure of the painting, on the one hand, we learn more about the intention of the artist itself, but we also learn more about how to restore it and how to conserve it. So the information that we obtain in these experiments also help conservators make sure that the paintings don't degrade, so that they can be preserved for future generations, so that the museum exhibition times increase, which then creates more revenue for the museum itself. So it has a whole ripple effect, why it's important. And this is quite funny because I had a conversation with Greg about some of the sample pre-processing steps over in the microscopy center. And it turns out that we embed a lot of samples in resin blocks. That's exactly the way that we prepare the painting cross-sections. So a conservator will use a scalpel, remove a very small piece, about 100 micrometers of a painting, and that we embed in usually in epoxy resin. Then we polish it from one side to expose the different layers of the stratigraphy. And then we have a flat surface that we can use for ToF-SIMS imaging. So it's actually a very similar way of pre-processing the samples. Yeah. Here's a SIMS image of lapis lazuli, and it's, at the bottom you can see the chemical composition, and by looking at different inorganic ions, we can see the structure of the lapis lazuli grains in this particular pigment. We can also compare different pigments and use that to determine their origin. So we can, for example, see this between saldonite and glauconite just by looking at different intensities for inorganic elements. And then we can apply that to the painting cross-section. So this is a painting cross-section where on the one hand we're having a look at carbon signals from bone black, signals from Naples yellow, signals from blue smalts, signals from the lead oxides, probably from lead white, aluminum from probably, I'm gonna guess, a lake pigment, and iron oxides from ground earth. If you want to know more about cultural heritage and SIMS, a couple of years ago, Caroline, together with me and Alain Brunel and Philippe Voltaire, published a tutorial in the Journal for Mass Spectrometry. So it gives a very nice overview of how SIMS can be used to investigate cultural heritage materials, specifically paintings. And Caroline and I, just before she joined the group, also published a book chapter on the examination of authenticity in artwork using all various types of mass spectrometry. So if anyone's interested in this kind of stuff, those are the papers and book chapters I would have a look at. I'll be quick. So the different parts of our project, she only started a couple of months ago. So first thing is to define reliable marker ions, do an optimization of the sacrificial samples. So we're also working with partners that can provide sacrificial samples before we move on to the real deal. And over there you can see the distribution of palmitic acid in a lead white layer. And an important problem that we have, because these resin blocks are not dielectric, we do have some charging issues when we try to extract the secondary ions. So she's also been working on how to overcome these challenges. Our two partners that provide historical samples are the Cure2 Institute of Art in London and the Rijksmuseum in Amsterdam. The Rijksmuseum is the National Museum of Amsterdam. And here are some examples of samples we get to look at. So from the Rijksmuseum right now we have some historical reconstructions that match some of Rembrandt's paintings, including the Night Watch. And we'll see how it goes, but if the measurements on the historical reconstructions go well, there is a potential to have a look at the Night Watch by Rembrandt. From the Cure2, we already got some samples from Westminster Abbey. So this over here is actually a cross-section from one of the wall paintings in Westminster Abbey. So that's where all the kings and queens of England are crowned. And like I said, one of the issues with these older samples, they've already been embedded, and they tend to embed in pretty big blocks. So there are some charging issues over there. But she's been working on some innovative ways to overcome them, and we just had some conversations about filing for a patent, so that's actually quite exciting. Oh yeah, and then the other part is, of course, apart from the actual science, is also involving the field of conservation. So we also work with Stroll, which is a local restoration company, and this is all about communicating these things between the analytical chemistry side and the cultural heritage side. So because this is multidisciplinary research, not everyone is aware of the capabilities on the other side. And we are going to make an active attempt during this project to really advertise the capabilities we have and what these types of technologies can bring to this field, because that's not done often enough. And I also see it in different fields. So when I talk to medical doctors, quite often they wish they had certain things so that they could see certain things, and then as an analytical chemist you tell them actually it exists. You simply haven't heard of it yet, and I haven't heard about your problem yet. So it's a matter of bringing all these people together, have a brainstorm, and then get these multidisciplinary projects jump-started. Yeah, and we're also working on open access database, and that's quite cool because we just opened it up with surface science spectra. So we're going to publish a pretty big collection of historical pigments using ToF-SIMS for use by other groups around the world. And we just published the very first paper of the open access database. And the next 2 have already been accepted, and the other 3 have been submitted. And I've been told that the QR code works, so if you scan the QR code, it should take you to the open access database. And also to everyone here, if you're going to be using SIMS in the future, SIMS, because of matrix effects and uncertainties in ion formation mechanisms, is very reliant on good reference spectrum. So I would encourage people to actually submit their spectral data to these reference databases to really help the field moving forward. Oh, and here are some other examples. This is actually something Kimberly did as an other side project. So she also had a look at a painting. This is not part of Caroline's project. This is a separate side project, but it quite nicely shows the type of resolution and type of ions that can be observed on these painting cross-sections. So she actually used the database of Caroline to look for the different clusters associated with a particular type of lead white, and actually found all the different clusters that were in Caroline's reference database. So that was a very nice immediate application of that published work. And using all the other elements that she can detect, she is also able to identify the other layers in this 3-layer system. And added to that, something that people might not know, because we're combining pigments that have heavy metals in them, like for example lead white, together with binders like oils. We do sometimes get formation of lead soaps in these historical paintings, especially after a long time. They form these little balls that get bigger and bigger, and they actually start pushing themselves through the varnish, damaging the painting. So conservators are very interested in learning more about how they are formed, why they are formed, how they can be stopped from forming, all those kinds of things. But they've never really been unambiguously identified, and she tested out the tandem MS feature on those spots containing lead soaps. And the lead soap is actually this, let's see, so this is the BB302 plus signal over here. So that's this one over here. And the lead soap is a teeny tiny peak of about 100 counts next to it. But by using the tandem MS feature, where we can purify our signal, removing any kind of background, we're able to create a tandem MS spectrum that nicely allows us to identify the lead soap for the first time. So this will soon be published as a technical note in JSMS, I believe. And this is the same thing, but for stearic acid instead of palmitic acid. So to conclude, we've gone over the different research lines in my group at the Maastricht University. So we had a look at a couple of pilot projects led by Kimberly on organometallic drugs and isotopic enrichment. We had a look at Maria's work on advanced ToF-SIMS image data analysis. We also had a look at the development of SIMS IHC and fast-pass microscopy IHC, on which Kimberly, Maria, and Edith are all working. And we had a look at the art research conducted by Caroline doing her Marie Curie. So what I hope you take away from this is that SIMS research can be quite diverse. It can be used for a very large number of interdisciplinary projects, and hopefully it gets everyone at Federal University of Minas Gerais excited for the arrival of the new SIMS instrument in the microscopy center. So let me quickly acknowledge all the members of the research group. Here they are during our away day in December. I also want to thank the master's students from HE Engineering that helped out with the data analysis project, my University of Maastricht colleagues, like Ron, Ian, Judith, and Bird. Eva, because she illustrated some of the illustrations that we used during the presentation, so we're also grateful to her. And of course also the external collaborators from Penn State, Ludwig Boltzmann, Strahl, Rijksmuseum, and Courtauld. And the following partners for sponsoring all the work. And if there are any questions, I'm very happy to answer them. Also very happy to answer fundamental questions on ToF-SIMS, but I figured for this type of presentation it was better to focus on the applications rather than the fundamentals. But if there are questions about the fundamentals, happy to answer them. Thank you very much.
a second.
Process pipeline on the third.
Yeah, very nice talk. I have a question along those lines in terms of data. There's someone that has some, computer science stuff here. So the question I have is for the AI processing, like PCA, K-means. Do you do that? I mean, how long do you train? Of course, that's unsupervised learning, right? But do you create your models and then you process them afterwards? And how long does it take once you do that? I mean, to process an image. Because it's a volume image, right? You have all the XY positions, all the coordinates, and then a spectra for each dot.
Yep.
And then how long does it take typically?
I'm going to give a very imprecise. Okay. I'm going to give a very imprecise answer. It depends. Depends on the size of the image, the number of pixels that we have, the richness of the mass spectra. So if you have a lot of peaks, that of course changes things a little bit. It depends on how much you downbin them. But I would say a single image, if you don't really downbin, you can probably still perform PCA, and I think it takes, I don't know, 10 minutes or something to get it processed.
10 minutes.
And the random forest, I did it on a small subset of 8 images with peak selection a couple of days ago, and that took me for 500 trees in the random forest about 2,000 seconds. And then 500 seconds for throwing new data down the tree. But it really depends on the pre-processing. That's really the key here, where if you use the entire mass spectral range without downbinning, you end up with very big data sets, and it becomes quite complicated. If you reduce it by downbinning or by peak picking, you immediately reduce the image size a little bit.
Okay, so the pre-process is manual? You have to all adjust everything back?
Yeah, so all the pre-processing is manual, and that's the part of the data analysis that takes the longest.
So there's no way you can do like a real-time processing, right? Because you still have that manual aspect that you have to take care of before feeding to the AI models.
Yeah, that's a big difficulty, and that's also something that one of my colleagues deals with, Eva Kuipers, who's interested in intraoperative diagnostics, but because she is working with in real-time processing. Yeah. Luckily, we're working with pathologists, so all the relevant humans are dead. So we can take our time with the analysis.
All right, thanks.
Hi, my question is about the possibility of imaging lipids. I think you said that it would be pretty straightforward to separate the different major classes of phospholipids in a sample.
Yeah, it is. And lipids also ionize very easily with these instruments, so much so that we jokingly call it a lipid microscope.
So would it be feasible then to look at the distribution of different classes of phospholipids in different regions of a cell, for example?
A single cell or a tissue?
Well, we could start with a tissue, but ideally.
Okay. Definitely in a tissue. Because in that case you can increase your pixel size a little bit, because one of the limiting factors is of course sensitivity. So if it is a very low abundant lipid, it might be more difficult to detect it. But generally we can see them, and even at the single cell level you can see intact lipids with this technique. And that's something I did during my PhD, where I did three-dimensional reconstructions of single neuronal cells. And in that case we could definitely see lipids in the cell membrane itself, and you could actually separate the cell membrane from the cytoplasm when we did the 3D sputtering.
And what about someday looking at differences in lipid composition between plasma membrane and other organelles?
I would say that's sort of at the limit of the capabilities of the technique at the moment, because we can bring it down to a spot size of about 100 nanometers. Currently, but the smaller the spot size, the less molecules you have to ionize, and the ionization efficiency isn't that great. We actually lose a lot more molecules. A lot more molecules are dissolved from the surface that stay neutral than that are actually charged. It's about 10 to the minus 5. So the smaller the spot size, which you need to have if you want to see those organelles, the less material you have to ionize, the more difficult it will be to detect it. So I feel that's sort of at the cutting edge of the technique at the moment. So we're moving in that direction, but I don't think we're quite there yet.
I am, of course, interested in the future of cationic analysis or proteins. And I expect that will probably come with new detectors, new Orbitraps, new material. Do you think that's in the near future or far future?
That is definitely in the near future because it already works with the nano-SIMS instruments. That has a DC beam. It has also been demonstrated with the J105 by Inoptica, which also uses a DC beam. I think the challenge with our instruments and those lanthanide-labeled antibodies is simply increasing the pulse width of the primary ion pulse. So I don't think there's a whole lot of instrumental development that's needed for that. I think we can make that work with the current instrumentation. But it's still relatively new, still needs to be developed, and that's what my group is currently working on. So hopefully between now and next year we can show you the protocols on how to do it.
Yeah, if there are other of course.
Two questions. Thank you. It was a very nice seminar. A question of somebody who knows something, but not really. Because would it be possible that in the same machine, because you showed very nice work on organic molecules, would it be possible, for example, on a geological sample monocyte, for example, in the same machine to identify which elements are there, or would it need to be a different machine?
No, those are things you can also investigate with this instrument because, like I said, it's good for elemental imaging and you can also see inorganic materials. That's why we use it on the painting samples. You just run into similar issues as with the painting samples in that geological samples are usually dielectric and they're not flat. And in this case, the samples need to be dielectric and flat. So I think that's more of an issue of sample prepping than it is of the instrument itself. But people do use it for geology, and I actually very briefly did a archaeology project. So in that case, we were looking at, I know it's not the same thing as a geological sample, but I would say close enough. And those were indeed the issues they were working with. So we had to do quite a bit of charge compensation and bias correction. But in the end, we were able to get some useful data out of those samples.
Nice. And the other question would be a little bit more on the collaboration issue, because we have a very nice group in Brazil which are working together as a net on cultural heritage, and we are turning into a strategic partner of the ARIS. I don't know if you
No.
know. Yeah, it's a collaboration of shared infrastructures in Europe, and we are seeing how we can set it up, and also working on databases. So maybe I get your contact by Greg and see, because there is a lot of things that we might be very interested in doing.
Well, and we're always interested in collaboration and sharing information. So yeah, let's talk about this after the seminar.
Brilliant. Thank you.
If anyone wants to talk to us tomorrow, Sebastiaan will be at the Center of Microscopy in the morning, so talk.
Sorry, I have another question. So how about like, say, for example, isotopic dating? Can you do that, like as geochronological studies and stuff?
Yes, in the sense that we can look at isotopes in materials, but there are more sensitive instruments for that. And especially for dating, you need to have that sensitivity and quantitative measurement. So I wouldn't, a SIMS instrument is not the thing that I would recommend for that type of research.
Okay, thanks everyone. We hope he comes back again to see us in the near future.
Pessoal, só para avisar, quem quiser certificado tem que pegar os QR-codes, tá? Para preencher o formulário aí, vamos receber por e-mail, tá bom? Tá aqui na parede, pode ficar em pé, tá? Obrigada.
Okay, we got it again, so.